Hello, this is Brian Kinghorn, and this is the fourth video in our series on genetic markers under mate cell, and we're looking at handling multiple recessive lethals and multiple gene edits. And the mutations behind these things can be SNP mutations, or they could be insertions, deletions, larger haplotypes, many possibilities there. And of course for gene edits we could be talking about knock-ins and knockouts. Now with many loci you can manage these one by one, and we've looked at that in a previous video. So in this example we've got 4 loci in 4 columns, and the top row shows the distributions for the favourable allele, and then for the 4 genotypic combinations down below we have distributions for those. So we can manage these individually, but it would become very tedious to do that for very many markers, and so it's good to have a method to handle these collectively. So tediousness is one problem, but probably a more important one is that the best way to handle a marker can depend on how you handle other markers, so interaction is the issue there. And with the example of multiple recessive lethals, well, you can only die once. That means that if you have the bad allele from father and the bad allele from mother at a single locus, then you're dead. And it doesn't really matter what happens at the other loci, that's not going to bring you back from the dead. If you're in the business of breeding animals for organ transplants and you're knocking out components that lead to tissue rejection, then one such component might not be acceptable, and therefore there's a lot of interaction involved in the same way there. On the other hand, if you're extracting genetic material from an extinct species in order to try and recreate that species, well, you're probably dealing with very many sites throughout the genome, many haplotypes, and there's plenty of opportunity for interactions there, and collective management of these effects is probably desirable. Basically, in any case where you've got nonlinearity and scale effects, then it's going to be difficult to handle markers one by one. So we have a mechanism to let make cell help you manage a group of markers collectively. So we currently use that mechanism to manage multiple lethal recessives. You can only die once. The other applications I alluded to here are a bit avant-garde, but there is some work underway and or on the horizon for these. However, for most gene edit work, you'll probably use one-by-one marker manipulation. So let's have a look at multiple lethal recessive loci and how we manage these collectively. There have been rather many projects in this area. Our experiences with a USDA project in beef cattle, where we were looking for missing homozygotes found typically through some sort of sequence data. So one develops a panel of many low frequency SNPs that are putatively recessive lethal, and basically that's because we're finding no homozygotes quite widely in the population and are therefore inferring that we have mortality in utero. So mate cell handles all these SNPs collectively as 2 traits. Lethal G, which is predicted mortality in the progeny, and that's basically looking at the genotypes that are predicted in the progeny, hence the G in lethal G. And lethal A for additive or average effects, looking for predicted mortality in later descendants. So let's have a look at this in mate cell. Here you can see the individual marker manipulation. We've actually got 100 marker loci in this example, and we're just looking at 6 of them here. And you can see that for marker 43, the prevalence of the favourable allele is not 100%, and so there is some evidence of a recessive lethal segregating for this particular marker. When we look under trait management, we see these 2 extra traits, lethal A and lethal G, and the 1 and the 100 are telling us that this is actually for markers 1 to 100 in the marker management tab. There may be other markers being managed individually. Let's look and see how we construct lethal A and lethal G. For any one mating, we're looking at the probability of mortality in the progeny from that mating. And that's this construct here. So this is mortality, and it is really constructed of 1 minus survival. It's easier to look just at survival. This P here is the probability that the sire has transmitted the bad allele at locus I. And likewise for the dam here. So if we have certainty that the bad allele has been transmitted from sire and dam, that's probability 1 times 1 equals 1. Then here we have 1 minus 1 is 0, and you're dead, because your survival is 0 and your mortality is 100%. And because this is a product rather than a summation, one element of 0 is enough to set the whole construct to 0 for survival or 1 for mortality. Now we come down here to look at probability of mortality in later descendants. Lethal A for alleles, or additivity perhaps. So the construct is very similar to what you see above, except instead of the sire, we've got what is essentially the predicted frequency of the bad allele in the progeny. So now we're looking at the grand progeny, that's the progeny of the progeny, and for the matings of those progeny we use the current population frequency, although if you did have some knowledge of where that was going, there would be the possibility, of course, of using a future frequency. But by and large, these frequencies will not change very much if you've got a large number of loci. It takes a lot to make changes in frequency. And later we'll look at swapping these constructs out for other constructs that reflect other target patterns of inheritance. But let's stick to the multiple lethal recessive loci and look at what happens in mate cell. So here we have our two traits, lethal A and lethal G, and the red ticks showing the current solution averages, the black the average of the candidates, and we have more or less the same means there, not much difference. Let's look at lethal G. I'll expand it. I'll ask to decrease mean and invoke that and resume. Now we'll get a change in scale probably here, but the key thing to notice is that the average has gone down considerably. So the probability of mortality on average has reduced considerably by putting a weighting of 1 in order to decrease the mean. Now this very favourable response is at some expense of being away from the frontier, so we'll moderate that. Let's put that weighting down to 1/4 and resume. And you can see that we've made considerable response in lowering mortality with very little change away from the frontier. And most of what you're seeing here is probably reallocation of mates in order to avoid mating heterozygotes for these very rare recessives, and therefore lowering the probability of getting affected animals and therefore lowering mortality. And if we look and see what's happened to lethal A, we haven't had quite so much response, just a little bit, which suggests that most of the response we're getting in lethal G is in fact due to mate allocation rather than selection. Here the selection effect is basically by changing allele frequencies, whereas here it's by changing mates to avoid mating of heterozygotes. Let's look at the pattern results you get for this sort of work, and this has been looked at in detail by Lindsay Upperman in this paper here, and this is the sort of thing that transpires. If we have embryo mortality, as it turns out to be, because we're looking at recessive lethals that we don't ever see in the live animal, we can move from something like 8% in these simulations down to close to 3% without any real change in the progeny index. That's largely through mate allocations, and then when we get further improvements in embryo mortality, we're changing the animals selected and therefore making compromises in progeny index. Before going on to consider collections of gene edits, let's just consider what we have with multiple lethal recessive loci. We have machinery to handle these collectively, and at the heart of that machinery are these two constructs, one for probability of mortality in progeny, which is short term, and one for later descendants, which is long term. So we can handle gene edits using the same machinery, but swapping these constructs out for what is appropriate for the pattern of inheritance in these gene edits. And we can have more than one inheritance target. We will talk about 2. And for each of these, we have a short-term and a long-term component, just as for the multiple lethal recessives. Now, the problem of multiple gene edits in a breeding program relates essentially to segregating gene edits. If you've been successful in site-specific insertion to create instant homozygotes, as a first order effect, that means that breeding the program is not going to be a problem. But of course that's not really true, because very often you will want to integrate those homozygotes into a wider population for production purposes. In many cases you'll be aiming for consistent products. That means that you will want to sell breeding products that are homozygous or not for gene edits and not heterozygous. That means that your clients can use these to create products that are indeed heterozygous in many cases, but they have the desired properties for the end user. And so it's likely that in many cases, segregation of gene edits will be an issue to be handled in the breeding program. Let's look at these inheritance targets, but not in too much detail. We're looking at summation of value across loci and genotypes. So V here is the value for the ith locus for the j-kth genotype, and we work out the probability of expression of that in the progeny. And that's essentially what this construct is here. And this is very similar, except the sire is replaced by the progeny, and we're using the population frequency to look further ahead. The value of this, of course, is that you can do pre-weighting of all your gene edit components and then manage these collectively, targeting either the progeny or in the long run, and have single weightings for each of these different components. And what I've called dominant lethal is an example of where you would not want a single instance of a non-edited allele at any locus, and that's the example we've had from xenotransplantation. In this case, you can see that if you had certainty of transmission of an unwanted allele from even just one parent, this would come to 0 and the whole construct would come to 0. And so that could be used to handle a whole collection. Of course, this may be a bit harsh and you might want to moderate such a function. And this is the sort of reason that we don't want to build these things into MateCell, because the constructs to use here depends very much on the situation. And so this is a case where functionality will be customized and invoked by agreement. So we're coming to the end of this video. You'll appreciate that much of what we're talking about, especially in relation to collections of gene edits, is largely for the future, but we're there and ready for that when it comes. The next video will talk about the genomic relationship matrix. Thank you for listening.